Good morning and welcome to the first set of the breakout sessions. We hope you enjoyed your break sponsored by Insmed. My name is Cassandra and I will be your room host throughout the 2 days for Track 3, the Drug Development Pathway, which is sponsored by PRA Health Sciences. We are excited that this track will also be live streamed. I would like to welcome our remote attendees to this session. As a reminder, we have our mobile app available, so be sure to download that, which we will be utilizing today for the Q&A session. Without further delay, I would like to introduce our first session, How a Therapy Gets Developed and Approved, the R&D Roadmap and the Growing Role of Patients. Please join me in welcoming Juliette Moritz, Executive Director of Strategic Development at Rare Disease Premier Research, and Alvin Shi, CEO of Enzivant.
Good morning, and thank you for joining us. We're going to take an approach to just sort of at a very high level review what a drug grows through as it works through clinical development and clinical approval. Then we're going to talk a little bit about how that pathway differs for some rare disease therapies. And then finally, we're going to go a little bit more in depth and look at different touch points in that process where patients and the patient experience can be shared with developers to help create a more patient-centric research experience. Okay. So we're going to talk a little bit, again, very high level, 3 main areas of research for a therapeutic: preclinical. That's when the therapeutic is tested in cells, tissues, and sometimes in animals, and that helps us characterize the mechanism of action or how the therapeutic actually works. Based on that data, we can then move into clinical trials, and these are controlled exposures in humans following protocols. They're very restricted in terms of inclusion and exclusion criteria so that we can be sure that when we get to the end of them, the data is clear and we're able to answer our research question. And then there are also some types of research that are done after the drug is actually approved for marketing, and that's called post-marketing surveillance, or in the case of a lot of rare research, post-marketing or post-approval requirements for additional testing once a drug has made it to the market. So there are 4 phases of clinical research. This is, again, when we're in humans. So phase 1 is the first time that a drug or therapy is tested in a human. These are very small trials, handful of patients, in the tens. We're looking mostly at safety in phase 1. These are often started in healthy, normal volunteers. And the goal is to try and understand exactly what the drug does in the body and what the body does to the drug. How is it broken down? How is it excreted? Those types of things we call pharmacodynamics and pharmacokinetics, and that's often the phase of the first time we're looking at the drug in humans. These usually last, they're short participation for an individual patient, but the entire phase 1 process can last a few months. And about 70% of the drugs that are tested in phase 1 are determined to be safe enough to move into phase 2. In phase 2, we're testing, again, safety. We test safety all the way through, but we're then also testing efficacy. So not only is it safe to give to people, but can we start to see whether it does what we think that it can do? And some of these phase 2 studies can include a placebo so that we're able to separate out whether what we're seeing in the patients is due to the drug or not. These are larger studies, sometimes can be in the 10s to 100, depending on the disease being studied. In a very common prevalent condition like diabetes or heart failure, these studies can be certainly into the hundreds. And a lot of times in phase 2, what we'll be doing is looking at different doses to see which dose makes the most sense, looks to be effective, and also the safest. 33% of the drugs that are tested in phase 2 make it into phase 3. So as you can see, this is a pretty severe kind of funneling when you're talking about anything that's coming out of the lab, making it into phase 3. Phase 3, again, is what we call in prevalent disease research pivotal trials. These are the trials upon which the safety and the efficacy are developed. They're tested in large populations of patients with the condition that you want to treat. And these can be hundreds of patients to thousands of patients. 70 to 90% of the drugs that make it this far will actually then go on to be submitted for review and approval by regulatory agencies. And as we mentioned, phase 4, post-marketing commitments in research for prevalent conditions are usually more about collecting real-world data. When you're doing phase 3, you're maybe excluding certain conditions that people might have or medications that they might be taking. Once the drug is approved for marketing, then you can go back and see how it works in those different settings. So just to give you a sense of how long the development cycle takes, this is for a prevalent condition, not a rare condition. Preclinical and drug discovery, you can start off with 10,000 compounds. This can take years just to get down to some compounds that are going to be worth taking into humans. Clinical research, again, is the part of the process where we're testing in humans. And out of the 10,000 that you started with, you may have about 5 that get all the way through preclinical and that are look promising enough to take into human testing. This process can take 6 to 8 years or more, depending on the therapy, the populations that are involved, and the condition that you're trying to treat. But out of those 5, maybe one of them makes it all the way through. So we're starting with 10,000 in the lab. We end up with 1 that makes it through the process. And then the review and approval portion of the whole cycle can actually take a significant amount of time as well. Depending on the regulatory pathway that's been approved for the drug and negotiated with the regulatory authorities, this process can take 6 months to a couple of years. And then after that, once the drug is actually determined to be safe and efficacious and approved to be sold, then we have to go through the process of having it approved for reimbursement by the payers. So when you hear an announcement that a drug has been approved or that the data has been really good and the company is going to submit it for approval, it's still probably some time before it's going to be on the market and available to patients in general. And as you can see, this data is, I think, from about 2015, and that says for a drug for a prevalent condition, it costs on average about $2.5 billion to take it all the way through from the lab to the market. So I'm going to turn it on over to Alvin now, and he's going to talk about how that process differs a little when we're looking at rare disease research.
Thanks, Juliet. My name's Alvin Shi. I'm a member of the Medical and Scientific Advisory Board for Global Genes. My day job is that I'm the CEO of a small rare disease biotech company based in Cambridge, Massachusetts called Enzivant. We're working on a couple of disease areas. The first is Farber disease, which is a rare lysosomal storage disease. And the other is complete DiGeorge syndrome, which is a primary immunodeficiency that results from genetic and other defects. So happy to tell you more about the company, but the primary goal today is to give you a little bit more insight as to the differences in rare disease clinical development. Now, Juliet gave you a really good overview of what the typical development pathway looks like. And I hope you have a good appreciation for the breadth and the complexity of the clinical development process. In rare diseases, there are some additional considerations that need to be taken into account. There are a number of different challenges. The first is that there are small populations that we're dealing with here. That goes without saying. You know, instead of looking at diabetes populations or heart disease populations, here we're looking at small populations, typically of genetic diseases. And so how do we find these patients for clinical trials, and how do they get enrolled and brought into the process? So the first and one of the key problems and issues that need to be addressed is how do we work within small populations of patients? And we'll get more into the complexity of that throughout the course of this dialogue. Secondly, these patients tend to be geographically dispersed. So all of the trials and all of the research that goes on is typically multinational in scope. And so how do we access those patients? How do we make sure that the research that we're doing is able to reach patients who are in different geographies? I'll give you one extreme example of that. In the research project that we're doing right now in Farber disease, we've discovered that there are actually a number of patients who are in Middle East countries, and in fact, some countries that are very, very difficult to access. So it's been incumbent on us to try and figure out a strategy to say, how do we get to these patients who could potentially benefit from being in a study so that we can actually discover and get the right geographic reach. Turns out to be a pretty complex issue because you're dealing not just with now scientific issues, but also geopolitical issues. The third issue right across the top is that oftentimes the sites that we're dealing with in doing clinical research can often be naive to research in general. And this is because a lot of times these are small sites. You might build a site around a particular patient cluster that you find. That site may or may not have experience in doing clinical research. And so this is a topic that's top of mind for a lot of us who run trials in rare disease. How do we actually enable these sites to do clinical research? And moving on to the second row, I'm sure this is something that many of you have probably experienced as well. There's oftentimes an unknown or sparse natural history set that you're working with. Now, this is important because whenever you want to measure the clinical impact of a therapeutic, you always want to understand what is the baseline natural history that you're comparing against. And oftentimes, that doesn't exist or is not in a robust enough format to be useful for a very robust clinical study. And so not having a natural history is something that would delay the clinical development process. Related to that, not having biomarkers can also be a very difficult hurdle to overcome. So in many diseases, you can look for a biomarker, which is usually a blood test, to show you how the disease is progressing and if your therapeutic is having some effect. Oftentimes in rare disease, that is not available because we simply don't have either the scientific knowledge or the patient base to fully explore and validate a biomarker. Same thing is the lack of surrogate endpoints. A surrogate endpoint is defined by regulatory agencies as being a surrogate or a biomarker or a clinical test that approximates the overall clinical path that the patient is undergoing. One of the more accepted ones is something like a 6-minute walk test. So this is just how far can a subject walk over the course of 6 minutes, and you track how that changes over the course of therapy. That's a relatively well-accepted surrogate endpoint. It's not valid for every disease. And so if you're working in a disease that doesn't have a surrogate endpoint, that makes it even more challenging to get that work started. And then obviously at the last piece, obstacles to patient retention. How do we make sure that all of the data that are collected are applicable and are able to be used for regulatory finding or regulatory filing? And this goes back to the fact that a lot of times these research sites can be naive. It also goes back to the fact that patient retention can be difficult, especially when you're talking about trials that can last multiple months, even years. So these are the challenges in rare disease research. Let me tell you about some of the ways that this manifests in the way that we think about clinical development paradigms in rare disease. The first is that rare disease trials don't always go through the standard pathway that we just talked about. Because the number of patients is small, it's incumbent on both patient populations and researchers to be as efficient as possible. And so what this means is that a lot of the effort that would normally be put into a phase 2 or even a phase 3 trial often gets front-loaded into either a phase 1 or a phase 1/2 trial. So you see here the nomenclature. Oftentimes things go from preclinical, which is, again, animal studies or studies in a petri dish or a plate, and they go directly into a phase 1/2. So what we mean here is that ordinarily you would go through a phase 1 in normal healthy volunteers and then do a phase 2 where you would find your dose in patients. Oftentimes those get mushed into one in rare disease settings. And that's because oftentimes the experience of a normal, healthy patient in a rare disease therapeutic isn't quite as meaningful as going directly into patients. So a good example of that is oftentimes we're working in lysosomal storage diseases or an enzyme deficiency, where the patient experience and what the drug does in that patient is going to be very different than what it does in a normal, healthy volunteer. And so oftentimes going into a phase 1 with normal, healthy volunteers just isn't that informative. So we really need to understand what it does in patients as soon as possible. So that's why you can see that oftentimes the phase 1 and phase 2 trials get mushed together. I'll say there are often times where even a phase 1/2/3 trial can be possible. And again, that depends on how much is understood about the disease and how many patients are available to begin working through that process. So, but there's a lot of flexibility, and there are a lot of ways in which the lines get blurred between phase 1, phase 2, and phase 3, because you really want to be able to assess efficacy as soon as it's possible, as soon as you have a good dose, and as soon as you're assured about the safety of the drug that's being tested. All of this flows into the last bucket, which is post-marketing. Oftentimes, if you're pursuing approval on the basis of a surrogate endpoint, the agency, either the US agency or the European agency, will often require that you do a confirmatory study to show that that surrogate endpoint correlates with clinical outcomes. And so this is what's referred to as a post-marketing or a phase 4 study. Those do occur in rare disease, especially in the cases where you have an accelerated approval on the basis of a surrogate endpoint. So what's the end result of all of this in terms of the impact on the way clinical trials look? What is the impact on timelines, for instance? And again, just to reinforce the concept here of kind of the ability for clinical research to be flexible in these stages. The proof of concept study, which is typically a phase 2 or 2B study in more prevalent conditions, can often occur in phase 1, as we talked about. So you'd be looking at a study that would be giving you your PK, which is your pharmacodynamics, how the drug behaves in the body, the PD, which is what the pharmacokinetics, the pharmacodynamics. All of this is telling you how the drug behaves in the body and what you can be looking for in terms of a therapeutic response. All of these things can be accomplished typically in an early stage study or a first-in-human study. So the way this manifests is that the trial profile, if you will, looks very different between rare disease and non-rare disease. This is some great research that Juliet and her group at Premier actually did, giving you a snapshot of both rare and non-rare diseases and how they differ in terms of the phases that each group is in. And you can see here the major differences on the rare disease side on the left. You can see that the pie is very much slanted towards the phase 2 part, whereas you see a lot less of that in non-rare disease. And so what you can take away from this is that there's a lot more early stage effort, and that the phase 1/2 and phase 2 tends to predominate, because again, that's where you would go as almost your first in human, and that's where you would go to look for your proof of concept. You can see that there's a lot less phase 4 work, because again, most times you will be looking to demonstrate efficacy during your phase 3 or during the phase 2/3. So you can see that the profiles are slightly different for non-rare versus rare. And so you might ask, because of this flexibility in the way that we can structure clinical research, does that necessarily mean that the timelines and the cost of doing clinical research in rare diseases is more efficient? And the answer is yes and no. It depends. Because there are a lot of considerations that go into designing rare disease clinical research. And there's a lot of complexity that isn't necessarily there when you're talking about more prevalent conditions. So in more prevalent conditions, there's often a very defined development process. There have been a lot of drugs that have already established the pathway towards approval. That's definitely not the case in rare diseases. For the most part, there might be 1 or 2 other trials that have been run in that disease space or a similar disease space. But for the most part, you're talking about really having to do that initial work to define what the development pathway looks like. And so that can add some time and complexity to the process. In prevalent conditions, you also have very accessible patient populations. Once you open a trial, you can have very good metrics around how long it's going to take you to enroll that trial, how long it's going to take you to get to a data readout on that trial. I would say in the rare disease space, it's much less predictable because, again, we're looking at very, very small patient populations. You're looking at sites that aren't necessarily geared towards rapid enrollment of subjects. And so this is a place where it can be highly variable in terms of the time that it takes to enroll patients, the cost of getting treatment to those patients, and the overall objective, which is to finish enrolling of that study and to get to a data readout. So all of these things lend to the complexity of doing rare disease clinical research, and it really reinforces the need for patients to be involved as early as possible, such that we can together overcome some of these hurdles and move more efficiently towards an approval. So with that, I'm going to turn it back over to Juliet, and she's going to walk through some of the detailed considerations.
Please stay up here because I'm
Sure.
sure you're going to have some relevant input as we walk through this. So now, again, very quick, high-level walkthrough of what drug development looks like for prevalent diseases, and then how that differs for rare diseases. Now we're going to go a little bit deeper and talk about individual points within the clinical development process where the patient voice and the patient experience can really impact how that development program is designed and executed. So in preclinical, again, this is when a company is in the lab. So they're testing in tissues, they're testing in animals, they're testing in cells, and they're hopefully thinking to themselves, I would need to know what it's like to live with this condition. I need to understand how this therapy might impact patients with this particular condition. But I don't know anybody that has this condition. It's not like diabetes where I can ask my mom or my aunt, you know. So how do I find out? And what kind of information would be helpful as I'm creating my development program? So when we're thinking about what kinds of information would be helpful to a drug developer in the preclinical phase, definitely just a better understanding of the disease burden or what it's like to live with this condition day in or day out, or to care for somebody with that condition day in and day out. As researchers, we often don't know. I mean, rare disease has an incredible component of folks who are involved because they know people, because they have folks in their families that have these conditions, but a number of us don't. And this information is invaluable. It's also really helpful to talk to patients and say, what are your goals for this treatment? We may not be able to offer a cure, but what symptoms would be meaningful to you for us to be able to impact with the therapy we're developing? What functional considerations are part of living with this condition that we could possibly address with the development of our therapy? As Dr. Shi had mentioned, what is the natural course of this condition? And so how do we understand that better so that then we're really better able to characterize whether our therapy is making a difference or not? And then again, the biomarkers, so the lab tests or different functional tests that'll help us measure meaningful outcomes for patients. So this is the kind of information that would be helpful. How do we as drug developers reach out to get that information? We can perform natural history studies. We can create or query existing patient registries. We can reach out to groups of patients and have discussions, focus groups. We can do online surveys through existing patient organizations. And we can reach out to advocacy organizations and have one-on-one discussions. But all of that information then feeds into what are we going to do with this therapy? Is this going to be a meaningful therapy to develop for this population? So each one of these points is an opportunity for a patient community to have some input. We're just going to talk really briefly. We've talked about registries, natural history studies, and then there are a very small subset of natural history studies that can be used to support regulatory submissions. So by definition, a registry is I never read slides, but I'm going to read this slide, so I apologize. An organized system for the collection, storage, retrieval, analysis, and dissemination of information on individual persons who either have a particular disease, condition or risk factor that predisposes them to the occurrence of a health-related event. These can be purpose-driven collection of demographic, epidemiological, so how often does this appear in the population, clinical effectiveness, so that also sometimes can help us identify how standard of care is working or not, cost effectiveness of existing therapies, quality of care, quality of life, and care pattern data, which can obviously shift depending on where you live and what your healthcare system is. So registries are sort of a broad group of the collection of all of these types of different data. Natural history studies actually document the natural course of a disease immediately prior to its inception, if you're lucky enough to capture that, progressing through the pre-symptomatic stage and the different clinical stages. And these are a little bit more rigid than a lot of registries. So they'll say, if you're participating in a natural history study, we're going to collect lab samples or imaging studies at certain points in time so that we can then compare that with other people in the study and really have a really good picture of how the disease progresses. And there are a very small subset of natural history studies that can actually support a regulatory submission by serving as a control group. I would caution everybody, don't move into a natural history study assuming it's going to be able to be used as a control group. That's going to have to definitely be discussed with the regulatory authorities, and they need to be conducted with the same rigor as a clinical trial. But it can happen when there are very, very small populations. But we just wanted to kind of clarify the vocabulary before we went much further. So the drug has made it out of the lab, and now we're thinking about testing in humans. So how can I, as a drug developer, develop a study where patients are going to want to participate and the information that we're going to get out of it is going to be meaningful and impactful to the patient population? So you can see our patient on the side, and he's saying, ask me about how to design this study. Ask me about how hard it would be for me to participate in this trial. Ask me about all of these different things, and I'm not going to talk about all of them on this slide because we go into a number of them on the next couple of slides, which are very data dense, but it's taking each of these and saying, what can a patient offer for all of these? But as you can see, there are a lot of opportunities for input here, and developers want to hear this input because ultimately we want to develop studies that are going to answer the research question and get a therapy to people with unmet medical need, but we can't do that if we don't design a study that we can enroll and where we can retain patients and get a data readout. So when we're talking about trial design, ask me about trial design. Reaching out to patients, patients can tell us, is this a realistic schedule of events? You want me coming into the clinic every 3 days? I can't really do that. So we need to find another way, or that's a study that'll be designed but never enrolled and never completed. I know here, when we're talking about time off of work or school, lengthy visits, impact on people's lives. I was working with a company one time, and they wanted to collect urine samples from patients, but they wanted to collect them 4 days in a row. And a number of the patients with this particular condition were still able to work, so they had jobs. So they were then put in the position of having to collect urine at their job and keep it and take it home and put it in their refrigerator and save it. Or worse, put it in work's refrigerator. So, and when I was talking to the medical officer at this company, he said to me, well, I don't see why this should be an issue. I would have no problem doing this. And I just, in my way, just said, have you asked a patient? Have you talked to somebody to see what this is like to have this condition, to know that their employer is probably already making accommodations for them, and then telling them they've got to carry around a jug of their urine with them for 4 days? So this is where that kind of interaction early on would have helped them design a better study. Or even if they had to have that criteria for scientific reasons, to help understand a better way to message it to the patients so that they could have buy-in on why it's so important. I'm not going to talk about every single one of these. This could be, a 3-day thing all by itself, but we just wanted to show you there are so many different opportunities for patients and their advocates to have input into how a trial is designed. Talking about the burden of participation. Again, as Dr. Shi said, patients are spread out all over the place. Participating in a rare disease trial means you might have to travel And if you're a parent with a child, you may have to deal with childcare, you may have to take time off of work, you may have other children that you need to deal with. So these are all things that we need to hear from the patient so that we can build a study that's going to be one that we can enroll and complete so that we can answer the question and move on to the next phase and hopefully have a successful development. Do you have any?
Yeah, so I would say, it's very important once you as a patient have a seat at the table, and we can talk more about how to get that seat at the table, but once you have that seat at the table, your input is critical to designing clinical research because you understand more than anybody else what the experience is like on a day-to-day basis. So one example of that is that we were trying to design a study where patients would be required to travel, and then on reaching the clinical site, they would be required to do a 6-minute walk test. Well, it turns out that if you're traveling from, and again, these are geographically dispersed patients, if you're spending a whole day in travel, and then someone sits you down and asks you to walk for 6 minutes, you're gonna be pretty tired. And that's gonna influence the way that you perform on that particular clinical outcome. And that's gonna have a dramatic impact on the way the clinical study reads out because your baseline measurements and all your subsequent measurements are going to be skewed. And so we decided, based on patient feedback, that travel would be a separate day and then all of the assessments would be run on a subsequent day, such that it was better for patients, also better for the trial. I would say the other place that you can plug in is in helping to determine some of the inclusion-exclusion criteria for the trials. Now, these are very, very important because this tells you exactly which patients are going to be eligible for a study and which patients are not going to be eligible for a study. And this is always a fine balance because as a clinical researcher, I want to ensure that there's some degree of homogeneity among the patients that we're looking at so that we have less variability in the data. But at the same time, we also want to ensure that there's broad access and that we're getting enough patients who will fit the criteria for the study. So this is a place where discussion with patient groups is very helpful in determining how wide do we want to cast our net, how do we want to draw the boundaries of this clinical study such that we can balance the need for patient homogeneity with the need to access a broad range of patients.
And I think following up on this last one for endpoint selection, this could be critical too because sometimes, and you'll see in the comment to the right, sometimes the regulatory agencies tell us flat out, you need to measure this, this, and this in order to get your drug approved. But sometimes there's some more wiggle room, or those may not be very well defined in rare diseases. So in that case, it makes sense to reach out to the patients and their caregivers and their advocates and say, what endpoints are going to be meaningful to you? Sometimes they can even be developed as a secondary endpoint.
Absolutely.
So it's not what drives the yes or no question on the therapeutic, but it helps us for the next time. I remember I had an experience where I was talking to a number of physicians who treat patients with a particular condition, and they all decided what would be a meaningful outcome or a meaningful endpoint for the patients, and the study was designed around that. But then when we had the opportunity to speak to patients, they came back and said, that doesn't matter to me at all. This is what's really more important to me. And we could have included that as a secondary endpoint so that we could have a better sense of what the approval of this therapeutic might mean for the patient population. Other opportunities are assessment selections. So, if you have a study where you are going to require patients to perform certain functional tests and that excludes a large number of that population, maybe that's not the assessment that you include. But that's a discussion to have with patients and then also with regulators. Trial awareness and messaging. This is huge. I mean, a lot of times getting the word out about a study can be sometimes challenging, but beyond that, it's more how do we do it? What's that message? Hey, we've got a study isn't always enough. We need to be more sensitive and understand what that trial opportunity offers to patients and their caregivers and their advocates. And we don't know that because we don't live it. So there's an opportunity for patients and advocates and caregivers to tell us how best to talk about the studies, how best to characterize the disease and what we're trying to do with our research. Same thing with study aid development. It's very helpful to have information from patients when we're developing patient manuals or even posters about the study. For you to tell us what's going to be most helpful for you in those materials. And if we design something and it doesn't make sense, can you help us figure out a way to say this that's going to be meaningful, that's going to help you be interested in participating in the study, help you understand what you're signing up for so that you stay in the study, and communicate effectively what our goals for the study are going to be. Informed consent and assent. Again, sometimes it's worth reaching out to patients and making sure that they understand exactly what it is that they're signing up for, and having that conversation, even maybe looking at a mock consent form or saying, if I said this way, would you really understand that I'm trying to say this? We are constrained in how we write consent forms and pediatric assent forms, but there is some opportunity for some wiggle room there, and only the patients can tell us what makes sense to the patients. Site recommendations, this often is something that we get from working with advocacy organizations who can tell us who are the investigators or physicians that treat a lot of these patients that are going to be able to really perform the protocol, and then advisory board participation. I don't know if you maybe might be able to speak to that in a little bit more detail.
Sure, absolutely. But before we get to advisory board participation, let me say a couple of words about site recommendations and the ability of patients to influence the way sites get selected. As Juliet mentioned, oftentimes patient advocacy groups will have preformed networks of investigators, and that is truly enabling for a drug company to come in and say, listen, I know that if I can get this through my preclinical studies and it shows good effect, that there's a network of investigators who are already prepared and have patients who could potentially be eligible for a study. Even better if that network has something like a common IRB that can eliminate some of the administrative upfront burden of setting up a study. So those are all things that can be very, very helpful and has largely been the domain of the larger patient advocacy groups. But I think we're starting to see that even smaller and smaller advocacy groups are really able to put together networks of investigators that can really enable research. And then in terms of ad boards, I think this is commonly the best way for people to get plugged in and to, like I mentioned earlier before, you have to get a seat at the table. And a lot of times the ad board is the initial way for companies to outreach and to put patients together in a non-threatening and collaborative environment to talk through the major issues that are coming up in the clinical development program and to work together to solve those issues. So ad boards are something that I, as a CEO, I will go to every patient ad board because that is where you hear exactly where the feedback is coming from. You get it directly from the patients. So if you're ever invited to one of these, do your best to be available. This is a place where really you get the most senior level people at the biotech or the pharma company who will come and listen. And they are there to ask questions, they're there to test ideas, but they're there to listen. And I will tell you, many times the outcome changes based on the discussion at these ad boards. So really give these some priority if you're able to make them.
I'm going to add a little caveat to Dr. Shee's comment. People who may have heard me speak before may have heard this point made before, but I think one of the things that industry can do better, besides just increasing our outreach in general to include the patient voice, is that when we gather that information, like through an ad board or a focus group or survey or outreach through an advocacy organization, we as industry need to do a better job of coming back and closing the loop with the people who participated in those conversations. We could do a better job of coming back and saying, thank you very much. We took everything you said. We changed our study here, but we weren't able to change it here, and here's why. So I can tell you that that's something that I talk about whenever I'm talking about rare disease research and trying to get researchers to involve patients and the patient voice is closing that loop. So this is not a one-way street. It's something that we can do better as well. All right, I'm going to let Dr. Shee talk a little bit more about the patient voice and the review and approval phase.
Yeah, so this is a very important topic. The FDA has come out very publicly and said that it is important for every trial as a primary endpoint to be able to demonstrate how that therapeutic affects the way a patient feels, functions, or survives. So that's very important language. They want to know how the therapeutic affects the way a patient feels, functions, or survives. And the only way to do that is to first understand what's important in the patient experience and then be able to demonstrate how that therapeutic affects those important things for those patients. So this is a place where I think, if anything, the role of the patient is going to increase over time. And this is a place where oftentimes we will look to bring patients with us into those dialogues with the FDA. Now, what that means is, I'm not just meeting you today and taking you with me to the FDA tomorrow. This means, there has to be a dialogue that's established such that we understand each other and that we're working together on this to ultimately get a therapeutic approved. And that's where all of our objectives are aligned. But this is something that should be expected in the future. I think you're going to see a lot more outreach from the FDA. I've had the FDA even directly call patients, not with me not there. This is something where they feel comfortable enough to even outreach to patients to understand how is it that this disease is affecting you? What would be a meaningful outcome in this clinical study? So I think you're going to start seeing a lot more of that.
Yeah, I'm sure most people in this room are familiar with or have at least heard the term 21st Century Cures or the 21st Century Cures Act, also known as just cures. And in that legislation, the FDA is actually directed in the next 5 years to come up with tools to incorporate patient engagement and the patient experience in most clinical trials. And then they're going to be developing metrics around how that happens. So this is something you're going to be hearing a lot more about. And hopefully this discussion today will introduce to you all of the different ways that, or some of the different ways that patient engagement and the patient experience can be included in the clinical trial development process and the clinical trial execution. But just something to prick up your ears about. It's going to be a couple years in development, but it's going to be something that's going to be much more common. And I, for one, am really glad to see that it's going to be mandated and at least standardized to some degree. But anyway.
Absolutely. So we want to make this as interactive as possible. I'm going to invite Cassandra to come back up. I know we have questions that have come through the mobile app, and we also have microphones set up in the room. So happy to take any questions.
Thank you, guys. So yes, please go ahead and feed your questions into the mobile app if you haven't, or come up to one of the mics and we will direct them accordingly. Go ahead.
Hi, I'm working on building websites and mobile apps for patient registries, mostly in a research context. And I'm wondering if you have any words of wisdom on how we can make those registries sort of as fluid or as interactive with sort of the inline pharma experience in drug development. So if you're looking for participants in registries, I'm wondering how we can make our registries more compatible with your process.
I think a lot of it has to do with the way it's developed. One of the biggest challenges even that pharma companies run into when we're developing registries is retrospective data and trying to go back into time and then going forward, and also how you're standardizing what kind of information you're collecting. So a lot of it has to do with exactly what assessments you're going to be including if you're capturing that information, how you're going to be documenting diagnosis of patients when they're entering the registry, whether you're collecting retrospective data, what that data looks like, because that can vary depending on the institution or the country, as well as missing data, because not every person is going to have the same data in the same way for every, for the entire course of their living with this condition. And so having those kinds of discussions and having, I would say, some outreach to really strong data managers that have pharma experience probably be helpful in the beginning, certainly.
Yeah, I would agree. And I think one of the key words that Juliet flashed up on the registry slide was that it's an organized system. And so the organization of it is critical in terms of saying, which are the data elements that we're going to capture, over what time period are these measurements useful. And then I would say the second thing is some ability to do source data verification. So if the ambition is to have the registry be useful for either post-marketing or filing purposes, then there has to be some element of source data verification that the agency or someone else could look at and say, okay, now I believe that this data is robust, and here's why.
So maybe uploading documents or something like that so that it's available for someone else to look at to reconcile against the data that's actually in the registry.
Great, thank you.
I'm going to pause here and go to one that came in via the app. So are you positive or negative about the new FDA commissioner's intent to modernize orphan drug development?
Yes.
I think the devil's in the details there.
Right. No, I think overall, there's been a lot of movement on the FDA's part to, especially in rare diseases, streamline the processes and make sure that it's reasonable what the agency is asking for. So I would say all in all, I've had very, very constructive relationships with the agency, and I think that that's going to continue. And I'll tell you about one of our programs. We actually have the only program that has been designated by the agency as both a breakthrough therapy as well as a regenerative medicine advanced therapy. And so we're the only known program, at least that I've heard of, that has both designations. And what that's allowed us to do is to have very open and constructive dialogue with the FDA. And we really feel like it's a true collaboration as opposed to, we sit on this side of the table, FDA sits on the other side. And it's been very, very constructive. So I think we're moving in a good direction.
Good.
Hi, my name is Romy. I am diagnosed with Lambert-Eaton myasthenia syndrome. I was misdiagnosed at first with MS and then finally diagnosed with LEMS. I was the first one on the clinical trial in Phoenix, Arizona at Barrows. So I'd fly from Philadelphia. I was one of those people that had to stay there for a couple months in a hotel. And one of the things I wanted to know is when they first started dosing me, after the first 20 minutes, I was able to walk for the first time in 8 years, pretty much. But then, since I was the first one on the trial, it was like they were dosing me up, and it wasn't until I was in the emergency room with AFib at like 250 heartbeats per minute when the doctor said to me, "You're going to come off the trial because of the side effect." I'm like, wait a second, you're dosing me up, and it's a speed effect. So when do you know enough is enough when you're starting a clinical trial? Although I was the first one, and now I'm a patient advocate, so when patients are starting on the expanded access program or on the clinical trial, I tell them, please start low, because they start having those heart palpitations and feel like those feelings that you're going to go into AFib, it's going to scare them, and they're not going to want to, take the drug anymore. So I feel like I'm the trendsetter because I was the first one, but I do all this with a little group of my LEMS friends that I have. And actually, one of the buddies here today. A lot of my little LEMS family are on twice the amount as I am. Although I'm the largest person, I'm on the smallest dosage, and I'm thinking that's because of the arrhythmia. But my doctor they were going to take me off the medication, and I said, no way, Jose, because you up this, and this is my threshold, I guess. But he made me get cardiac release. So I had to fly back to Philadelphia and get cardiac catheterization very quickly to come back to stay on the trial.
Wow.
So, I just didn't, are there thresholds where they know, this is good enough? I felt like he was going to keep going up to 100 milligrams a day, and that could have just taken me out.
Right. And this, again, is part of the difficulty of doing studies in rare disease. And first, thank you for your participation in that study.
Yeah, I did
it's
a
very
couple
courageous.
times.
There's always a first patient treated, and in this case,
I was
that was
number
you.
one.
That's amazing. What I would say is, obviously, every drug has to go through a battery of preclinical tests where they try to assess the safety, and they also try to calculate what's called a human equivalent dose. So you take the animals and you translate the doses that are given to those animals into a dose range for people. And then you typically, over the course of phase 1/2 trials, go up on that dose to understand where you hit toxicity. Because ultimately, you want to have the best chance of getting remission from your disease, which means you want to hit the optimal dose.
Right.
It's usually not going to be the first one, may or may not be the second one. And that's why they typically do this dose escalation scheme.
Right.
But really, the only way to understand that in patients is to run into some sort of dose-limiting toxicity. And so in many cases, what we'll have is an ability to go down from that dose to a dose that's safe for you, And then that
Exactly.
becomes the dose that we try to get people to. So that
Exactly.
really is the only way of doing it. It's an empirical process.
Yeah, so it was interesting because I don't know if you know, it was Dr. Siva Kumar and Dr. Lada at Barrows. But I started off with one medication, then went on to the other, had better results. But the second doctor was pushing the threshold. But at the end of the day, now I'm at a good place. I'm not as high as I need to be, but walking is a privilege, so
Well,
at the end of the
and-
day, I'm at a better place.
And the drug behaves differently patient to patient.
Right.
So it's
Exactly.
not, we're all made differently. And so what we try to do is enable the best dose range. But oftentimes it takes experimentation to figure out, how the dose should ultimately be given to patients.
I'll make one comment, and then I think we need to move on. But
Okay.
one of the other things that we were talking about in terms of informed consent, especially in these first-in-human trials, is to make sure that we're very clear about exactly what's going to happen, that the dose is going to escalate, what happens when you hit a dose where you're starting to see adverse effects or evidence of toxicity, and making sure that the patients actually truly understand what that looks like before they get there.
You
So
know
that-
what? That was not explained to me. I mean, now that you're saying it, it totally makes sense, but that was not explained to me. And also, I think I was in a desperate situation to try some kind of alternative that would work.
Sure.
But I was not told about all the side effects when you go up. So that
Well,
was a good learning.
oftentimes in early studies, we don't even know.
Yeah, like
what
I wrote the side
can
effects
be.
possibly. So.
Right, so that all comes with experience oftentimes.
Okay, thank you.
Thank you
Thank
so
you.
much. So I know we've got a line here, so I'm going to offer up their expertise to maybe touch base with you guys after this, because we have time for one more question. So I'm going to take one from the app here. So what is a crossover study, and how much does it help?
Do you want to go first?
Sure. Crossover studies are actually extremely helpful in rare disease because we're talking about very limited patient populations. So if there's only 60 to 100 patients known in the world with a particular condition, it's really hard to define a study that's going to have patients on therapy and patients not on therapy against whom you can compare the effect, hopefully, of the therapeutic. So a crossover study is a study where patients actually serve as their own controls within the trial. So at one point, they either start off not on the therapy and they're observed for a certain period of time, and then they're offered the therapy and observed for a certain period of time, or they could be put on the therapy for a certain period of time and then taken off and then followed. Most of the time that's blinded so that the patient doesn't know which phase they're in, but that allows us to gather twice the data from the same number of patients.
Right, so it's a way of potentially increasing power because you have your placebo arm that will cross over to a treatment arm, and you have your treatment arm that crosses over to placebo. You might ask, though, why aren't all trials designed this way? There are some liabilities to crossovers as well.
Yeah.
And part of it is that you could have a lingering drug effect, and you don't know what the washout period is. So in some ways, it's not as clean as doing a straight, randomized, double-blind, placebo-controlled study, because you have crossover effects of patients who were on therapy that then go to placebo. You don't know how long the effect of that drug is lingering after they move on to a placebo arm. So there are definitely pluses and minuses. Overall, I think it's a very useful tool that
Yeah.
should be explored, but it's not always appropriate in every situation.
Great, thank you. Well, thank you, Juliet and Elvin. Let's give them one round of applause for their great presentation. At this time, if you can jump onto the mobile app and take the brief survey, we will go ahead and end this session and allow you some time to transition to the next breakout session. Thank you for joining us.